All right. Hi, everybody. Sorry for being a few minutes late. Just needed to finish digesting my lunch. So, probably best thing to do is spend a few moments, if people need it, to talk about the lab report that's due tonight. Some of you have taken the opportunity to get first drafts to me, and I've sent those back with comments and stuff. If you haven't, now would be a good time to ask about stuff because that's what will make your lab report better. Bada. Good question, Rose. No, there isn't any penalty for going longer than 3 pages. It's basically as long as it needs to be is the easy answer. And so if it takes you 3 or 4 pages to say what you want to say, then that's fine. You're not going to get more points for writing more words, but you do need to make sure that everything that I've asked of you is in your lab report because that's what you'll be getting graded on. Yeah, I'd rather have a nice big graph that I can actually read without squinting, and stuff like that than have everything squished down to fit into an arbitrary volume. Questions? And you can interrupt with questions too. It's just kind of easier to do them with all at once if you have them. Okay, so again, remember, you should include the formulas for chi-square. So yes, for chi-square, odds ratio, and map distance. And so some of those are easier to put as actual equations, even if you just copy and paste a picture from the web. Some of them you can type them out, like map distance is, number of recombinants divided by the total number of progeny, for example. You could write that as an equation, or you could just describe what you did. But either way, those are part of your methods. So you shouldn't write, that's in the methods. But you shouldn't write out or you don't need to write out the math for how you calculated your chi-square value. Right, so you don't have to kind of write out all of the observes and the expected squared divided by expected and, kind of all of that stuff, because I'm just going to assume that you actually did that, right, to get your chi-squared value. You don't need to show me that you did it, but you do need to tell me how you did it, which is what the methods are for. Something that also has popped up in a few people's rough drafts is putting the results in the methods section and putting the methods in the results section. Those two are pretty clearly delineated, right? The methods is where you describe what you did, right? I did this back cross. This back cross was done by crossing a homozygous dominant to a homozygous recessive, taking the heterozygous offspring and crossing those back to the homozygous recessive male, right? I scored the 4 different phenotype combinations and their frequencies, right? That's methods. The actual counts for the 4 different phenotype combinations, or even those by sex, that is in your results as a table. Similarly, in the results, you don't describe how you did the chi-squared calculation because that's what should be in methods, because that's a method. But you do record the chi-square value, the degrees of freedom, and the p-value, because those are your results, right? That's the analysis of your results. So that's something to be very kind of careful about. Methods describe what you did. Results describe what you found. Discussion, summarize your results. Well, repeat your hypothesis for a start, summarize your results, and then interpret what those results mean. So each section has its purpose. Another thing that's also, did that help, Rose? Good. What was the other thing? Oh yeah. So few students have had some confusion between the hypothesis and the predictions they're testing with the chi-square. So I've had some where the student hypothesizes that these two genes are linked, right? So that there is no recombination going on between them, which is a perfectly fine hypothesis, right? But then they carry out their chi-square with an equal number of the 4 different phenotype combinations being their expected values, right? So if the total is 2000, the expected values they use is 500, 500, 500, 500, right? So those expected values come from your predictions, which come from your hypothesis. So if you're hypothesizing that your 2 genes are totally linked, then your prediction is that there is no recombination between them. If there's no recombination, you'd expect to have 1000 one parental combination, 1000 of a different parental combination, 0 recombinants. That's your chi-squared test, right? So the chi-squared test is at the end of a long series of kind of thought processes, which start with, what's my hypothesis? What do I think is going on? If my hypothesis is correct, what would I predict that I would see? Right? And from my predictions, I can work out my expected values for a chi-squared test. Right? So those all have to match. You can't predict total linkage and no recombination in your introduction and then do a chi-squared test which tests the hypothesis that you're having no linkage in your results and then come back and say that your hypothesis was supported, because you're mixing different things. You have to be consistent the whole way through. So that's something else that's kind of popped up in the rough drafts that I've seen. Trying to think of anything else kind of in general. Can't really think of much else really. Oh, another one that has come up a few times is when you're talking about linkage, what we're talking about is linkage between genes. But linkage essentially results in, right, equals different allele combinations being found more commonly together. That's essentially what the odds ratio is testing. E.g., if you have A, you'll have capital B. And if you have A, you'll have B. That's what we mean by alleles traveling together during linkage. Right, so in a sense, what you can do is if you find allele, uppercase A, you can make a pretty strong prediction that you'll also find uppercase B, further down the chromosome. Because those two travel together, you're much, much less likely to find a lowercase b for that gene B. Right, so that's a little bit of kind of language that some people are getting a little bit tripped up on. Linkage is between genes. The result of linkage is some allele combinations of those two genes being found more commonly than others. It's kind of a subtle thing, but it's an important one. I think those are the main issues that kind of come up. Also, don't forget, people generally put like two out of three of those things for the discussion. Not all of them. You need all of them. Okay, does anyone have any other questions about the lab report? That's correct, Adam. By tonight, by email, please. And with your name in the file name too. Just makes my life so much easier. I don't have to save things. Change the file name and stuff, which gets a bit of a drag when you have 20 of them to do. All right, cool. Okay, so changing gears. Essentially what we've done so far in our FlyDNA project is we really just talked about the two different phenotypes, right? The yellow versus brown body, like the molecular basis of that. And so, typically once you have kind of isolated a mutant, you want to figure out, well, where is that mutation? What gene is it in? Will the identity or sequence of that gene tell me some more about what is normally done, or will the fact that it's not working and gives me this particular phenotype give me a function for that gene, right? So yellow is a great example of that because, when it's not functioning, you get less pigmented flies. And so the phenotype of a yellow mutant is less pigment. And so that tells you normally, like the opposite, is that the yellow gene is involved in pigment production somehow, right? So what we want to do now is we want to go, okay, well, what is the molecular basis of that lack of pigment, right? And in many cases, if you're actually isolating this mutant yourself, you'd want to figure out what the sequence of that gene is, because that might actually tell you something about what that gene does or how it does it and how its lack of activity results in that phenotype. And so the very, very first step of any of this stuff is getting the DNA out of the organism. And typically organisms don't like giving you their DNA, right? So you have to smush them up. So for, soft and squishy stuff, we're talking about flies here. Humans are pretty soft and squishy as well, same as mice. It's actually pretty easy, right? You just kind of like pound the snot out of it in a mortar and pestle. For other organisms like the ones I work on, which have a thick cuticle, or insects which have a carapace and exoskeleton, things like that, they're actually much, much harder to get open. And so for my worms, I have to freeze thaw them in liquid nitrogen to crack open their little wormy bodies so I can get the stuff inside. So generally speaking, there's going to be some kind of mechanical step. Yeah, let's bring up the right, and so this is called the homogenization step. So really what that word means is making everything the same, right? And so if you have a tissue sample of, say, liver, right, you want to make it a uniform paste or slurry of stuff, cells. So really you want to break down the kind of the physical structure of that tissue. Working with flies, you want to break down the flies. And so, you'd end up with a little tube full of like fly heads and wings and legs and bits of thorax and exoskeleton and that stuff, right? And so the purpose of that is to kind of just like mix it all up. But it's also, you need to make sure that the chemicals you add later have access to all of those cells, right? So for example, if you took a single fly, you put it in the lysis mixture, only the cells that are available to that lysis mixture, which are like the eyes, some of the abdomen, whatever, would get broken open. And that's not very efficient. Even more so with worms, like their cuticle protects almost the whole of their body. And so you wouldn't get much kind of in exchange, right? So if you want to lyse open all of those cells, or as many as possible, you just have to break them open. And, sometimes that's done mechanically, so we have like little mortar and pestles that fit into tubes. Sometimes you can put them through what's called a French press, which isn't for making coffee. It's like a hydraulic ram which squeezes them through a small hole, which obviously they don't appreciate. Or you can do it by freeze thawing, which is or even enzymatically as well. That's a little less common. But the main purpose is to kind of break down that tissue so that all those cells are now kind of exposed, I guess, to whatever's going to happen next. And so the next thing is that you have, all those cells, and you have a plasma membrane, which is a phospholipid bilayer. You've got a whole load of other junk, and you have another phospholipid bilayer, which contains the DNA. That's the nuclear membrane, right? So you have to break those open to be able to get that DNA out, right? And so this is, essentially, this is like a cooking recipe, right? Of, how to get the DNA out. And the best way of doing that is by disrupting those membranes, right? And so you can use something like Dawn, right, which is a detergent. And detergents, really what detergents do is they break up fat, right? That's kind of their main role. And so we use something called SDS, or sodium dodecyl sulfate, which is another one. You can use some nasty ass ones like sodium hydroxide, which is also known as lye or drain cleaner. That's nasty. I don't know if you've ever got it on your fingers, but it makes your fingers feel soapy. It's because basically it's breaking down the skin cells on your fingers so they're nice like oil, right, back and forth. It's also according to Breaking Bad anyway, great for disposing of bodies as well. So there you go. Very, very effective detergent. Yeah, again, it's not, I mean, I use it for metalworking. It's really good for taking the anodized layer off of aluminium. And that stuff, once it gets going, it's just nasty. Like the fumes that come off of it, like you don't want to be doing that in an enclosed space. And it's a very exothermic reaction. So it kind of makes the solution really hot. Yeah, don't do it in the same, like if you're just in like a one room apartment where your bathroom's in the same room as where you sleep, you know, just leave. It's produces some really nasty chemicals. Or better still, don't wash your, you know, your hair off your brush down the sink. That's what I tell my kids. Because honestly, having three females in the household all with long hair is not a lot of fun if you're the one that cleans out the drains all the time. But that's probably more than what you needed to know, right? Anyway, so the whole purpose of the lysis step is to break down all those membranes and allow that DNA to come out, right? So that it's in solution. The thing is, now you have essentially a soup of, you know, you're going to have fragments of plasma membrane or just phospholipid bilayer. You're going to have organelles of all kinds of different shapes and sizes, mitochondria, ER. You're going to have proteins, you're going to have RNA. And what else is attached to that DNA? Actually, we learned it in class this morning. What macromolecule is DNA wrapped around? I think. What's chromatin made up of? DNA and histones, proteins, right? So if you want to get nice clean DNA out, you got to get rid of the protein too and all that other jazz. So sometimes we do an RNA degradation step. It's typically good if you want to kind of do any kind of DNA sequencing, right? You want to get rid of RNA because it will just, you know, interfere with stuff. But whatever you want to use this DNA for, you have to get rid of that protein because if that protein remains stuck to that DNA, it's going to stop you from doing PCR. It's going to stop you from doing any kind of sequencing or enzymatic digestion. It's just going to get in the way. So once you've made that kind of slurry or soup of cell contents, the next step is to just degrade the protein. Right, and I mean, the proteins are going to be partly denatured anyway by what are called chaotropic salts. I actually looked up what that means this morning. It's kind of fun. What they do is they interfere. And they denature protein. If I can spell nature. So that's part of kind of the mix that's added essentially to this lysis process. But there will also still be a lot of protein complex to the DNA, right? Because it's kind of all wrapped up and it's tightly structured. And so before we even want to try and get the DNA out, we have to degrade the protein. And that's what this protein degradation step is all about. And essentially that involves adding a non-specific proteinase called proteinase K. There. And then you just leave it for a while. And so that proteinase K, you know, it kind of floats around. Every time it finds some protein, it will chews it up. If it's a histone, it will chew it up. If it's a, I don't know, ribosome, it will chew it up. It will just, you know, non-specifically chew up any protein that it finds. And so once you've done all of that, now you have still a slurry or soup of stuff, all those different bits and pieces, right? And you have DNA that is on its own, right? Still within that mix, but it's now like naked DNA, I guess one could say, right? But it's still within all of that soup. And in that soup are ions and protein fragments and RNA molecules and just stuff that will massively interfere with anything that you want to do with that DNA afterwards. Right. So up until this point, the whole emphasis has been how do we get that DNA out of the cell in a state which we can purify it? That's what the homogenization, lysis, and degradation steps are all about. But even if you do all of those, you still have all those other macromolecules and stuff, junk almost, kicking around, which you do not want with your DNA. You want to get your DNA as pure as you can. Right. And so there are different ways of getting that DNA out and leaving all the other stuff behind. Right. There's an old school kind of, wow, shoot, what's that? It's in Triazole. Nasty, nasty, nasty. Oh, I just can't remember off the top of my head. Yeah, doesn't matter. But there's like old school ethanol precipitations, right, where DNA is kind of insoluble in ethanol, high enough ethanol, so you could do that. You could use something called Triazole, which is a, oh, there you go, phenol. How could I forget? You could do a phenol chloroform extraction, and that stuff is nasty too, right? So that will make you feel all kinds of, yeah, woozy-headed and pie-eyed if you do enough of it. Have to do it in a fume cupboard or fume hood for safety. Or you could kind of take the easy route and you could use what are called DNA purification columns, right? And so these are really cool. I'd love using these things. So basically, purification. These are often called mini columns. And what they consist of they kind of basically look like that, right? So this is a column. This is our collection tube. And this bit here is a silica layer, right? And the cool thing about this is it binds DNA selectively. At a specific pH. So as long as you make sure that your, let's just make that a little bit bigger. Your cell slurry is in the correct pH. I can't remember if it's, I think it's slightly acidic, but I can't remember off the top of my head. You put your cell slurry in here, you spin it in a centrifuge, that will result in all of the stuff you don't want, also known as junk, ending up in the bottom, and then your DNA stays stuck to the filter. So once that DNA is stuck there, then you just wash the DNA a bunch of times to get rid of all the other nasties that you added to help the process, such as chaotropic salts, right? Which are really useful for breaking things open, but you don't want them hanging around in your DNA afterwards. And you do all of that, get everything nice and clean. I'm just going to take a picture of this. And then once you've done that, all you have to do is change the pH. And so if you now put neutral pH, it's called a elution buffer in the top. The DNA that's in the filter, the silica filter, now it dissociates, right? It kind of just lifts up off of the filter. You spin it again. This is called the elution step. And now you'll end up with your liquid, which has your DNA in it at the bottom. So the main purpose of this is it isolates the DNA out of the general mix of stuff that the DNA was originally in, all the other cellular components. You just throw those away, toss them out. You can allows you to work with this DNA while it's immobilized on that filter for a bit. And then after adding your elution buffer, that DNA lifts off, and now you can spin it and collect it at the bottom of the collection tube. Then once you have that, your DNA is actually usually pretty clean. You know, it'll be free of most proteins, most RNAs, and typically free of all the salts as well, because those are cleaned off during the wash step. And so once you have that DNA, then you can do all kinds of different stuff with it, right? You can use it for PCR, you can use it for genome sequencing, you can use it for cloning, you can chop it up in a specific way if you wish to. It's totally up to you. But the aim in the game is to get that DNA in as pure a form as possible, because all the other stuff that's present in the cell or even in the cleaning process can screw up with any of those downstream processes. And so you want to get that DNA out as clean as possible. It's kind of a balance, right? Because you want to get it really clean, but you will need enough of it to use it for something, right? And so this process, this is this kind of mini column deal, pardon me, is very handy. It's very quick. It's very safe as well. You don't have to deal with like really nasty chemicals like phenol and chloroform and stuff. And it generates pretty clean DNA. What it doesn't do is generate lots of DNA, right? So this is great if you need some DNA, but not a lot of it. If you need a lot of really high quality DNA, you'd probably use a different, more time consuming and perhaps more perilous approach. But this is very effective for what we would use it for. And so essentially that's the next step in the process. What we'll be doing in lab on Thursday is going through how you can figure out whether or not you have good quality DNA. How much DNA do you have? Are there any contaminants that we need to worry about? Stuff like that. First step though is just getting the DNA out, and that's really what this is all about. So the assignment on Blackboard is really just going through, you know, these different steps, what order do they occur in. It's all based off of the information that's present in this handout and what we talked about today. And then there's just a couple of questions like, you know, why would you want to do this? Like, what kinds of uses can you think of for this DNA, for example? And that's pretty much it. Nothing much more to it than that. Cool. Does anybody have any questions? I have a couple of emails to respond to. So if you haven't seen, you sent them to me late last night or this morning. You haven't seen a response. It's just because I haven't got around to it yet. But I will do as soon as I've finished with this lot. Cool beans. Okay, so if you have any questions about the assignment or anything like that, we can talk about that on Thursday, or you can email me in the meantime as well. But that's all I have to share. Has anybody got any questions about what we covered today or about the report for tonight? No. All right. How about that for a result? You actually finished before lab is supposed to start. I should get a tip for that, right? I can pass around the hat or something. Cool. Okay. Well, good luck everybody in the exam. There is a file which exams due by midnight tomorrow night. 7th, midnight of the 7th. So you have all day today or all day tomorrow, depending on what else is going on in your life, to be able to take the exam.
Excuse me, sir.
Mm-hmm.
For some reason when it said that you just added the test, it says that it's due the 17th.
Oh yeah, so I might need to figure that out. So that's actually, I'll have to double check all of that. There's a good reason for it. So the reason is that one of our students has got deployed on short notice. He's in the military and he's coming back on the 16th and he wasn't able. Yeah, no. He wasn't able to take it before he was deployed. So I need to figure that out so that all you lot don't have until the 17th to take it, but he does. The main reason is the results are made available after the due date. And so I think the availability date is just until tomorrow night. So even if, you know, you're like, ha ha ha, I don't have to take it until the 17th, you wouldn't find it. So there.
Yeah, that makes sense. I think that's why I got confused with the first exam.
Yeah.
I probably didn't listen correctly. And I saw that. So
yeah, so if I like to leave the due date a little bit open, because if people have trouble with like internet connection or whatever and they can't take the exam when they're supposed to, it's still available for them to take, but they don't have all the answers, right? And so that's why there's like a window after each exam when people can make it up if they didn't take it. But after the due date's passed and those exam answers become available, then you know you can't really take it. So that's why. So there's a reason for it. It's not like I'm completely bonkers or anything. But it should only be available until tomorrow midnight.
okay, yeah, that makes sense. That's understandable. Thank you.
Yeah, no worries. It's just one of those oddities, that's all. Cool. All right, everybody. Good luck in the exam. Also, there's a second file with the 3-point cross data in it, which should also be available in the exam folder that the exam 2 is in. You'll need to use that for answering some of the questions. That has essentially what similar to what we worked out this morning on it. So, you know, maybe if I were to give you like some tips, maybe perhaps have a look at that first. I didn't tell you that. Oh shit, this is going to be online. Oh well, maybe I did, but have a look at that first. That might save you a little bit of time. There you go. Cool. All right, I shall see you all on Thursday, if not. And as always, let me know if you have any questions, okay? Bye-bye.